our second session. A little housekeeping before we get started with the lecture today. We have a newfangled white card system that we're going to try today for the first time. So on the way out, everybody please wipe your card in. We're going to plug it into the computer and it will capture your ID number. For those of you who are not actually registered for the course, we also need you to sign in on a sheet to give us your name and your ID number because we didn't have a way to capture that before. And so after today, everybody will just swipe. Does that make sense? There you go. Okay, our lecturer today is Dr. Prescott Deininger. He received his AB in chemistry from the University of California at Santa Cruz and his PhD from UC Davis. His graduate work concentrated on the sequence organization of the human genome. He was a postdoc at UC San Diego. Then he was a NATO fellow at the Medical Research Council in Cambridge, England, where he worked with Dr. Frederick Sanger, who invented DNA sequencing and won 2 Nobel Prizes in chemistry. While working in Cambridge, Dr. Deininger invented the procedure currently in use for shotgun sequencing of genomes. Here at Tulane, he is the Joe W. and Dorothy Brown Chair in Oncology, a professor of epidemiology in the School of Public Health and Tropical Medicine, and director of the Tulane Cancer Center. He also serves as the co-director of the Louisiana Cancer Research Consortium. Dr. Deininger is a fellow of the American Association for the Advancement of Science. He served as a journal editor on numerous editorial boards and on many, many study sections. Now, I have 2 fun facts to share with you about Dr. Deininger today.
Which ones did you pick?
He met the Emperor of Japan when he trained with a Japan Prize winner. And he published an article in the magazine Fine Woodworking about how to make an oversized, wide-body wooden tennis racket. So please join me in welcoming Dr. Deininger.
With a Japanese master.
No, I trained with Ted Friedman at UC San Diego, who first conceptualized gene therapy. Okay. He won the Japan Prize, which is their attempt to compete with the Nobel Prize.
Right. Please welcome
Dr. Deininger. But he was my sensei after that. Well, thank you. That was a lot of stuff I didn't even tell you about there. So very nice. So we're going to talk about sort of data and data integrity and what your responsibilities are, what my responsibilities are, and so on with data. It's the heart of what we do as scientists. And so basically we have this implicit contract with scientists and with the rest of the population that we will show good integrity with everything. And you know, it's not only important that we do good science and are honest in our publications so that we don't mess up other scientists and so on, but you know, every time you see something in the news where somebody has done something unethical or inappropriate in science, it weakens our whole field. It weakens the trust of society in us. And so it's a very, very important feature. An awful lot of what I'm going to tell you about is common sense, but you know, sometimes we haven't thought about some of these things yet. And so you have to look at some of the questions to figure it out. So we're going to talk a little bit about some of the issues in data acquisition, to keep objectivity. We're going to talk about the lab notebook, which is probably one of the weakest spots anywhere as far as keeping things right. We'll talk a little bit about data errors and a little bit about the ownership of the data. So it's really important that we not only do good experiments, but that we record them well and that we keep our data well. And proper acquisition and record keeping is the foundation of everything we're going to do in the future. If you don't have a record of what you did, for all intents and purposes, you didn't really do it, at least not from anybody else's perspective. We need this for basically for future experiments. We need it for patents and inventions. And you know, all I can tell you is, you think, oh, I just did this experiment experiment. I've done this experiment 20 times now. I know everything about this experiment. So that if I just remember a couple words about it, it'll tell me exactly what I did. Well, it turns out 3 months or 6 months later, or 2 years later, that's not at all true. Okay, I can't tell you how many times I've gone back and scratched my head over my own notebook trying to figure out, much less somebody else coming into my notebook and trying to figure out. So it really pays, and we'll talk about some details on that. And as you do any science, it's important that we have quality data. We need to be quantitative. And we have different kinds of data. You have your raw data, which might be something as simple as, a note in your notebook about counting up number of cells or something like that. It could also be instrument output. In fact, this is becoming more and more true. In my field, we get many gigabytes, even terabytes of DNA sequence all the time. You have to figure out, how do I make sure that data, not only that it's there for me to analyze, but that I don't lose the raw data, because if I lose that raw data, then all my analysis is worthless because I can't back it up. Okay, so you have to figure out how to back up your data. You have to figure, and how to maintain all of this data appropriately. Some of it can go in the notebook. Some of it can't go in your notebook. You got to figure out how to connect your notebook, though, to wherever the data instrument readings are. Okay. And then, of course, you have process data. That's not anything. Okay. You have process data, charts, analyze data. One thing I'm going to tell you, putting a lot of this stuff into your notebook is a really good thing, because you really, as you're going into keeping track of your data, your thought processes are really important, going from raw data forward to the charts and the graphs. And then there's all the little things that you ought to pay attention to. A little note, something that didn't quite go right. I dropped this on the bench top before I put it back in the test tube, or anything like that, or anything you can think that might affect the experiment, anything that is not written out clearly in a standard protocol, you might need to make a note of. You want to put pictures of things whenever possible, movie slides, whatever other thing. Then there's this one other kind of data that we don't really think of being data, but it is. It's some sort of biological specimen. That means, for instance, if I do an experiment on a tumor sample, maybe on the DNA or whatever, that turns that DNA sort of into data because I've published on it. Somebody else might want to do another experiment on the same sample, maybe do the RNA analysis on that sample, or maybe look for a protein in that same sample, or some other thing, or tie that data together. So you actually have to take care of your samples. In fact, I sat on a committee recently from the National Academy of Sciences, which was how to protect data from disasters. Okay, and we have experience here in New Orleans with disaster. We've been through Katrina, where we lost a lot of biological samples, and it was a disaster for careers. So taking care of that part of your data is really important. I up at NYU, where they've also had trouble, there were people there who had done all this great analysis, and then they couldn't finish because they couldn't do the next step on them because they lost the biological samples, and they had to start from scratch. Okay, so you got to take care of all forms of your data. Now, objectivity. I mean, as scientists, I think we probably all think of ourselves as being objective, but there are all kinds of little impartial biases we can have. Okay, we have to make sure that in no way are we motivated by personal gain in what we're doing. And we have to make sure that we have a hypothesis and we do a rigorous test of the hypothesis. One thing I've been found a few times is you can't be too married to your hypothesis. Okay? You got to be willing to disprove your hypothesis. In fact, a sign of a sign of a bad scientist when I'm reviewing a grant is somebody who says, my hypothesis is this. And from then on, the assumption is the hypothesis is right in every experiment. Okay, they never tell me what results might actually tell them, oh, that's not true. Okay, so always face the fact that your hypothesis might be wrong. That's the whole point of a hypothesis. Is it's a great idea. It fits the current facts, but you're going to test that hypothesis and maybe it'll be different. And certainly in my career, there's many times when I probably believed too strongly. Like I remember when RNA splicing came along, which you guys all take for granted, but that was totally radical. And I said, that can't be true. Well, we always learn DNA to RNA to protein. That's it. Okay, nobody put splicing. And then, well, when they started doing splicing, I was very skeptical, and I was wrong. Okay, so you gotta adjust, and I have adjusted and now studied splicing many times, but you have to accept that you're not always right. And that's exactly what I'm talking about here. Is don't get too attached to your hypothesis. The alternative hypothesis may be even more interesting. So, what kind of personal gain is there? Okay. One example is your desire to please your supervisor or your mentor. Okay. We'll have an example that illustrates one of those sorts of things. I've had students who did an experiment multiple times because they thought they were getting the wrong answer because it wasn't what they were expecting to get, and I think they were afraid they'd disappoint me if that was the case. You don't want to be biased by what you think somebody wants you to tell them. You just have to generate the data. Obviously, at various stages, you're affected by the thoughts of advancing in your career. You have some data, and some of the data doesn't fit this wonderful hypothesis. Do you ignore that data because maybe it's bad data, or do you accept that it might be there and not publish your cell paper, not get promoted or whatever? So you have to, it's very important that we not get too caught up in that we have to get a certain result and because it'll be really good for us. Obviously, some kinds of data will improve your chance of getting a grant, and ignoring other types of data that go against your hypothesis. All of these are, basically things that would help you advance, and you have to not let that happen. And we have a lot of structures to avoid that, but again, you can't always have structures to avoid that. You will be faced with many times where you have to think about, the disappointment of having a nice project collapse because the data don't support where you're going. Same thing for publication. If you have too many contradictory experiments, publication either goes way down in quality or shouldn't be out there at all. Okay, so your data is data. I love data. I always tell my students, there's no such thing as bad data. It's just data that doesn't prove what you thought it was going to prove. But I can tell you a few times, when I was a grad student, my wife had already moved on to San Diego. I had a postdoc all lined up. I was doing the controls for an experiment, and they didn't look right. Okay, so turned out it took me another 6 months to finish to figure that out, but it got me a really important paper because the controls were showing me actually something. I had to find controls for my controls, and there was an observation there. But so data is great. Okay, so here's a case study. A PI gives a theory to a new grad student. The grad student acquires the data to confirm or refute the theory. The PI explains their anticipation of finding experimental data having certain values. The graduate student generates experimental data with approximately the values. The data are published. The PI garners recognition and glory and tenure. A grant is renewed. The graduate student gets a PhD. Everybody's happy, right? And then the problem is, and this is something you really have to watch out for. The next student comes in, starts working on the project, and has difficulties reproducing the data. And what I have, I can remember a really clear story of this where with one of my colleagues, a student came in and just kept pounding away and couldn't reproduce some data. They worked for years trying to prove something, to further something that probably wasn't right. So it's very important that you, as a scientist, even if it's in the same lab, don't take everything for gospel that was done in the past. Keep an open mind, and if your data are pointing a different way, you better catch it, or you can waste an awful lot of time trying to further a result that wasn't quite right. And after further investigation, they found that the first graduate student chose, in certain cases where there were ambiguous values, he chose the ones that were closer to the PI's expectations. So here, trying to please the PI, trying to get a good result. And so that emphasized the trend in the data that wasn't really necessarily there in the full data. And I think this was one of the first questions, wasn't it? Okay, so here's the questions for you guys to consider. Well, at least try the Vox.
Okay, so everyone take out your cell phone or your laptop and please log into live.voxvote.com.
Did you guys do this last time?
Yeah.
Oh.
And so when you get there, you're going to put in the pin you see on the screen, which is 99082.
There you go, live.voxvote.com. We'll see how much time all of this takes. That's my only concern with this. Live.vox, V-O-X-V-O-T-E. And then 99802. Okay, and so should the PI have restrained herself from mentioning his or her anticipations? Just like in that example before. So I guess we have a yes or no answer. As you guys get into it, give me your answers. And at some point it will start summing them up. Nobody's in there yet, it looks like. There we go. Is that it? Yes. Okay. Fair enough. Well, so this gives us a general idea. We got 35 voters already. So 60% say yes, 40% say no. Now, probably 100% of you are right, because it depends on exactly how you put the emphasis on this question. Be a lousy test question, okay? Should they have restrained themselves? Here's the way I look at it. A PI should talk to the student about their hypothesis. And they should talk to the student about what the data might look like if that hypothesis is right. There's nothing wrong with that at all. What's wrong would be if they in any way, if they don't also emphasize the fact that the hypothesis may be wrong. In fact, it would be a really good exercise on the PI's part to say, you know what, if this other possibility is right, an alternative hypothesis is right, then the data might look like this. So the main error the PI made was perhaps not doing a good enough job to make the student realize that the hypothesis, he wasn't married to the hypothesis, and that there were alternatives. On the other hand, the student, the misbehavior was probably mostly on the student's part, in the sense the student should never have arbitrarily tossed out data. Okay. The end result, though, is the student. They both are damaged by that, right? The student has now published a paper that's got bad stuff in it. The PI, people in the field are going to recognize that they maybe also published something that wasn't good. Now, that's assuming it ever gets noticed. There's an awful lot of bad data out there, but still, it can and often is noticed. And the higher the profile something is, the more often it's noticed. Let's see, do I have another question in this of these? Which was the second question?
That one.
Okay. Oops.
Should the PI have more closely supervised the acquisition?
Yes. And we've already been voting. You guys are getting way up there. That's good. Okay. And I think, so anybody who says no, want to say why they feel no? I don't want to put anybody on the spot, because one way to look at it might be ultimately the student should be honest, and they've cheated their data a little bit. But I think most of us would recognize that it falls on a PI to train students. Now, ultimately, a PI could have said everything right, and the student might still cheat because they want to get ahead for personal gain, right? Get a better paper or whatever.
Well, it also could have been pretty innocent, couldn't it? You're going to round up or round down. Yeah.
You'd have to look at that. And again, the whole question is whether it was done deliberately. Mm-hmm. And there are some things. So, for instance, I used to do cell cycle experiments, and they involved labeling cells, and you'd have to take fields in a microscope and count. Mm-hmm. Well, it would be great if the cells were black and white after the experiment, but there's various shades of gray in the whole process. Okay, so you count in fields, and you have to say this one replicated and this one didn't. You can't say this one had a little bit of replication. Okay, so you're counting, and the question is where do you draw the line? You see a lot of this kind of stuff in pathology when you do analysis, where you're giving it a grade of a 1, 2, 3, or 4 in terms of the amount of staining. And so the question is, if I know my sample has that I want replication to have been increased, I might, when I'm looking at that sample, cheat it up a little bit with just naturally. Mm-hmm. And then on the controls, I'm a little bit more rigorous about my cutoff. So you have to watch that sort of thing, and there's tricks for that. You count your samples before you know who the control and the experiment are, so that you're not biased. Or what we used to do is have somebody else in the lab count the same sample to see whether we all got the same result. Should the PI have insisted on a more thorough check of data reproducibility? This I think is absolutely. Do we have that? I cut this out of the question because I didn't want to take up too much time on this. But I think there's no question about this. I've seen things where the PI likes to only see a final product from a student. That's bad. Okay, the PI needs to be looking at the notebook, needs to be looking at the raw data, and teaching the student how to analyze it, and making sure that they're not taking data. There are reasons you can take out data points, but you need to make sure that you use the right reasons for it, and that's the PI's job to teach them. So the PI has to be aware. When I was first training students, I would reread all of their sequencing gels initially until I knew that they knew how to read a sequencing gel and they got accurate results. Sometimes you have to do that. Either that, when you're away from the lab as a PI, you might have to have somebody reproduce a small chunk of their data as well to really convince yourself that something's right. And now what should the PI do? This is an interesting question. What should that PI do?
Retract and do the experiment again.
Yeah, I think the question here would be that's probably the most rigorous of the answers. You might say, well, I'm not quite sure that I was wrong, depending on how it was, and just redo the whole experiment first. But yes, you have the obligation to retract if you find out that it was falsified. And that retraction might only be one figure, and it might not affect all of the paper, but you have to publish a correction at the very least about it. Does that happen all the time? Only when people get caught, pretty much. Okay. It's hard to bring yourself to do it. What I will tell you, I've seen a few cases of outright fraud in science. I went on sabbatical up at Harvard, and they just a year or two before gone through a case where somebody really falsified data. And what the PI did was he did it right. He just pretty much said, we can't trust anything this guy did. They either reproduced things or retracted things right away. And it actually ended up not hurting that PI. Whereas if you fight it, if you say, oh, no, well, our stuff's all fine, it goes on for years where people keep publishing, oh, this is wrong, this is wrong, this is wrong. That can lead to a problem. And it can also lead to punishments if it's in the most egregious kind of case. Case study 2. PI must supervise the design of experiments in the process of acquiring and storing data. Accordingly, this is the answers. So the PI must acknowledge some negligence, and because we can't contaminate the literature with incorrect information. And so this is the general suggestion. There's one way or the other. You have to confirm whether you're right or wrong, and retract anything that's not right. Case study 2. A PI acquires data that portray a wonderful correlation between the administration of a drug and a physiological response. About 10% of the data, however, lie far removed from the predicted values. There are explanations as to why these data are different. For example, some experimental parameters have not been well controlled and were different for the experiments in which the results deviate. The PI chooses to ignore the outlying data in the publication. Was that okay?
No.
It's not absolutely cut and dried, and because there are some rules for ignoring data, outliers. Well, again, I'd have to say, well, if he eliminated the data because they didn't fit his hypothesis, he's absolutely wrong. Okay, there's no question about it. And if there's an objective reason to discount the data, preferably something that was noted before you found the data was bad. Okay, like I dropped this sample on the floor and picked it up, and it was so valuable I decided I'd finish the experiment. Okay, or anything like that, or that my equipment went bad, my incubator, if we had a power outage and my incubator went down for 6 hours. Okay. But then you should take any data that came from that kind of experiment, from that was affected by that particular event, and throw it out. But if there are those kinds of justifications, they're really clear, then it's okay to do it, I think. But again, as a scientist, you have to really make sure you're not saying, well, those data came from when there were a lot of sunspots, and I think that, because you could look around for something that might have been going on. You don't want to do that.
What would you say is an acceptable tolerance for outliers? Because 10% sounds high.
Well, there's various rules of thumb. You have a data point in an experiment that goes like 2 standard deviations away from the rest of the data. Mm-hmm. There's a rule of thumb that's often considered an outlier.
Right.
Okay. Other than that, like I say, you really want to think, well, if I have noisy data, was there anything that I did in the experiment that screwed it up? And figure out who your core best data is.
Right.
And analyze it. But again, it might mean that you have to repeat the experiment so you have enough repetitions to give yourself good statistics. I don't think there's a fixed number, because a whole lot. Some experiments are just incredibly accurate and reproducible. Others are almost sloppy by their nature and need a lot of repetition to get good, clean data. Okay, the notebook. I think probably most of us as PIs, this is one of the places we fail the most. It's because it's kind of boring to teach people notebooks, okay? And we kind of very often depend on other people in the lab to teach the new guys how to do a notebook or whatever. But the notebook is really, really, really important, okay? And it should be a bound, numbered pages. You're not allowed to remove anything from a notebook once it goes in. Okay, no tearing out pages, no erasing. You may put X's through something, especially if you make a note why you put an X through it. Like, I didn't finish this experiment because I got drunk and fell asleep. Each experimental day you should have a date on it so we know when an experiment took place. Okay. And it always annoyed me because I was always keeping 4 or 5 experiments going at once. That you'd then, by an experiment that took multiple days, you'd have these experiments intermingled with one another in your notebook. Okay, now maybe what you could do is keep a separate notebook for each kind of experiment if that bothers you. But one way or the other, you really need to everything dated, and as long as you label which experiment you're working on, you can separate them out. I can't emphasize how much some of this kind of stuff. So why? Put some of your thought processes in your notebook. It's not a dry thing where you just put numbers down. Say, my hypothesis is, so I chose to do this kind of an experiment. And similarly, later on, when you look at your results, put a few of your thoughts about the interpretation of the results. Because when you come back to look at the numbers later, you may not really remember exactly what you were thinking. And it really helps. You need a description of the experiment itself. Now, sometimes you'll have some standard protocols somewhere, in which case it's nice to have those in some also bound things so you can refer back to that standard protocol. So you don't want to write your protocol out completely every time, but you do need to make sure that if it's a standard protocol, and of course if you deviate from that standard protocol, you really need to write it out. You should, as much as possible, have your raw data in there or the location of the raw data. And like I say, descriptions of anything that goes wrong, any of your conclusions, those sorts of things. And it's a good idea to say, oh, I got this result. I think next we should check such and such, because that will really help somebody who's looking through your notebook, including yourself a couple of years later, say, that's why they did that experiment. Okay. Now, I'm going to mention here electronic notebooks. For about 2 years now, we've looked around at electronic notebooks and thought about electronic notebooks. I think in principle, they're a great idea. For those who don't know, they actually take care of most of this sort of stuff. They won't allow you to delete raw data. Once it goes in, it's good. There's all kinds of free ones out there, free up to a point at least. We've looked at it as a university a little bit, just figuring out what the right one to be. We haven't quite sorted it out, but it wouldn't be a bad idea. There are notebooks where the PI can actually look at your notebook and so on. Can't mess with it, and people can not share a notebook, but at least people can share access to a notebook, see different things. All kinds of great ideas. I'd love if anybody's exploring these and wants to do them. Some of you young computer people who do everything on the computer anyway might actually really love to do this. We haven't quite figured out what the best one is yet for us. And the right notebook, unfortunately, might be different. For instance, some of them have tools for DNA kind of analysis linked to the notebook, which is great. But some people don't do a lot of DNA type of analysis. They might need they might have more fun with a different variant. Okay, let's see what we got. We got a case study 3 on the notebook. A graduate student begins a research project and discusses the project thoroughly with his dissertation advisor. And the student does his reading. The advisor doesn't mention laboratory notebooks. Okay. So the student begins the work and begins to get great results. And the advisor discusses the results with them periodically, and everybody's excited. And after about 6 months, they're ready to write it up. And the student brings all these great graphs and tables and text for the paper to the PI. Okay, and I've seen a lot of labs that actually function this way. The problem is, upon studying the draft, the advisor has some questions about, some of the data that went in and wants to see the notebook. And what he finds is that there is no notebook. There is a bunch of paper towels with notations on them in the individual sheets and some other scribblings in a drawer. No dates. Some can't even be found at all. Okay. Did I leave this as a question or not? I can't remember. Who's responsible? Here we go, guys. Okay, well, I just added the both answer this time, and that's obviously whatever. Everybody's a little bit responsible, obviously. The PI needs to. Do I have the answers here? Yeah. Well, and so they both share the responsibility, and then the question is what should be done. We have a start over. Any other answers? I don't think there really is another answer. If you don't have a notebook, the data don't exist. Now, the only good thing about an experiment that you do like that is it probably gives you some idea that you're going to be successful, and you know how to do it now, so you can repeat the data quicker. But there's no question that until you get into a notebook, and the date for the experiment will now be whenever it's done. So if somebody else did it a month before you might have lost your patent rights to it meanwhile. And it was the advisor's responsibility to make sure that the student had a notebook and understood how to use it. But the student will bear the brunt of most of the pain of this. They'll have to redo it. They've lost 6 months of what is a very short career already. Hopefully it will still be longer, but still, the earlier you are in your career, the more that time loss feels painful, right? Whereas a PI might have 4 other people working away doing stuff, making a notebook, so they're making progress in other ways, so it's only a small proportion. Whereas for that student, it's very important. So, as a student, as much as you can, you need to take responsibility for yourself, even if it means asking your PI, how should I be doing this? Now, here's an example of what I thought was an okay notebook. It has 2 features that like it'll have a full experiment drawn out here. It's hard to see some of this, but like they'll have some data. They label their data so you can tell. Because a lot of time you might think, oh, I always load my gels such and such a way, and they'll just keep pasting them in there, and you won't bother to label what the lanes are. Well, it turns out a year from now you may not remember what the lanes were or whatever. And well, you can't really make it, but there are some nice comments in here. I don't know if I can see them better on my screen here. Well, saying what it's a Hilda-Dose curve on something here, and it says here try transforming, which is suggesting a new experiment from it or whatever. This is a decent notebook, obviously, and I can actually read the handwriting at this. And it could be a little bit better, but this would be probably a typical standard for a notebook. The more you can put into it, though, the better. I guess one of the benefits of an electronic notebook is you don't have to deal with handwriting issues. Now, here's another notebook. Who wants to explain this experiment to me? Okay. And it went on. There was a bunch of experiments. I can tell the date, at least, they got that much right. Here's some data. I think probably that number tells me something about where the number of the electronic file, not where it is. But I don't know what those lanes are. I don't even know where the top of the gel is on that, okay? So this is just to show you how bad things can be. So it really is worth taking a little extra time to create a notebook, a good notebook. Now let's talk about scientific errors. You can skew your data by poor scientific design. You can, at the experiment level, ignoring some data can change things. You can screw up the analysis by a poor statistical treatment or forcing data together in artificial ways that, there's something in the genomics world where we deal with multiple testing. The idea when you do 1 million experiments, that you can always find something interesting out of 1 million data points to cluster together, but it could be chance, not. So if you take all of your data and just look for something that looks like a cluster, you'll probably find some clusters. Whether or not they can repeat or whether or not they're real is another story. There's interpretation, which is the question of whether you have a personal bias. Do you actually consider, when you're doing an experiment, when you have a hypothesis, you need to consider all alternatives. And sometimes maybe we don't even know what all the alternatives is, because before we knew about splicing, I didn't know I had to consider alternative splicing. And there might be, so you have to just be aware, look very broadly at possible interpretations. And then there's fraud, okay? The key here is fraud requires the intent to deceive. If you have, this is very bad, by the way. That's why there's the death head there. If you have the intent to deceive, it takes it up from being a bad scientist to being a crook. Okay, and there are consequences. There are way worse consequences here, which include being thrown out of graduate school. I've seen examples of people who were banned from getting NIH grants for 3 or 5 years. Loss of tenure, being fired. All of those things can happen for fraud. So, sampling error. Sometimes it's just too small a sample. You got to make sure. Again, that's why you work. And I've learned over the years, it's always good to work with a statistician and to work with them before you generate the data, so that they tell you whether your experimental design will generate enough data to do the job right. Then there's a selection bias thing, distorting the results by how you do it. This is an interesting example, taking a poll in front of the Democratic National Headquarters about what you think of Trump. Okay, that would be an example where you might get a bias. It might be correct.
There's an implicit bias in almost all poll numbers because they all call home phones. How many people have home phones anymore?
I do. It's from my
But a lot of people
No, you're
don't.
right.
And that's
Well, and there's a lot of us who won't answer our phone if they're not in our. So who knows about polls? But the same is true with other kinds of experiments too. There's all kinds of ways if you can bias it in terms of how you set up the experiment. There can be measurement error. How many of you have your pipetment recalibrated every 6 months? How many of you knew that you had to recalibrate pipetment? I always hated when I have to pay that bill, and everybody brings out like 50 pipetment out on the bench top, and I got to pay $29 each to have them recalibrated. But at least once in a while, they should be, if not recalibrated, at least tested for accuracy. Okay, and the same is true for most experiments. This classification, I don't remember what we're dealing with there. Confounding error, uncontrolled variables, things that we talked about before where something goes wrong with an experiment in the middle of it that wasn't intended, like an incubator power going off, incubator running dry. If you're doing a circadian rhythm experiment and some idiot flicks on the light in the room and the melatonin all dissolves right away. Okay, and then there's phases of the moon and sunspots not being controlled properly. But anyway, all kinds of confounding error. You got to keep your eye open for that as best you can. But that's why we repeat experiments and require independent repetitions. And then fraud. So all of these kinds of mistakes, bad experimental design, lousy notebooks, any of that stuff is not fraud. Not unless you are deliberately cheating and making up data for experiments you didn't do, saying you did repetitions when you didn't, changing data of any type, only throwing away data you don't like, or just intentionally misinterpreting data. All of those are fraud. All of those are very bad. So I just there's a site, the government, NIH has all kinds of stuff. You won't be able to read this very well, but it talks about what you can do to your data. Looks a lot better on my computer. I can't even read that hardly. What I will tell you is you have to treat an image as data, which means you have to store your images in a way where they're backed up and where they're tied to your notebook in some way. And that's actually a little tricky because sometimes the computer will go down where the data was originally stored. If you didn't put it away somewhere in a backup, you could lose data. You should keep the original, even if you are going to do some legal manipulation of the data, like cropping or something like that. You need to keep the original handy. You're allowed to make simple adjustments. What's a simple adjustment? A simple adjustment would be something like cropping, only showing the lanes that are relevant. Okay. And you can also sort of like change the intensity up and down as long as you do the whole image. What you're not allowed to do is change the intensity in one region and leave the intensity different in another, unless you label it, unless you put say, hey, draw an arrow and say this lane I'm showing darker for a specific reason by such and such. But you can make some data manipulations as long as you explain them very carefully and why they were done. Okay, you can do a lot of things where you do the entire image, but doing pieces needs to be very carefully labeled. Be careful about filtering your data. Okay, you need again, if you filter your data, explain exactly why. Sometimes copying data degrades it, so be careful about using copies, Xeroxes of a film that you then take a picture with your iPhone and so on. Intensity measurements. Actually, intensity measurements are something you have to be very careful about. I mean, how many people know that film is nowhere near linear, that a band's intensity isn't directly proportional to the signal? Film has this threshold where you have to get up to a certain intensity before you see anything. Then it's linear over a very short range, and then it's saturated. So some of the new electronic instruments are way more accurate, and you can do that. But the other thing I've always struggled with is how do you subtract background on films in the image? Because sometimes the background is different in different places. You have to think that through. Why is there more background in the lane? Should I be using the stuff outside the lane or in the lane as the background? I can't give you a good answer. You need to work that out with your PI and figure out what the right answers are. But you have to be very careful about the intensity measurements. Compression loses data, and another obvious one is you have to make sure that you're careful with your magnifications. You can't show one thing at one magnification, another at another, and try to compare them directly. So you got to tell what the magnifications are, and usually make sure you're showing a 20x image on both of them if you're going to try to compare them. And then and resizing an image in terms of pixels is a lot like cloning, that it can degrade. You can lose image quality. So you have to be careful with all of those things. We do an awful lot of electronic stuff. This website is where that came from. So it's a great reference to look at some real-world examples of fraud that are detected. And what I'll tell you is there's a lot of cases where people are actually reading somebody's papers. And just by looking at the data, they can see it's been manipulated or reused in unusual ways and things like that. So, if somebody's really interested in your work, they're likely to be looking at your multiple papers and comparing them. And there are real-world examples of people screwing up their data deliberately. And this one also gives some of these general suggestions about what is appropriate for data manipulation. So I just have a few examples I wanted to show you. Here's a published figure. You guys probably have seen this kind of figure in the molecular biology world where we deal with bands a lot. People don't want to show the whole gel. They're often showing, in theory, for a certain sample, they'll show, say, in this case, I think it's a Western blot or various proteins that are expressed. This is your control. Okay. You look at this and you say, okay, this is a little ugly here. But in a paper where these things are often made really small, glitches sometimes get by that shouldn't. Okay, when you look at some of the raw data, this is actually the raw data for this. Okay, so now, as somebody who's looked at a lot of gels, first thing I see is, you notice it's not all clean bands. There's this splotchiness. That's an air bubble. Those are probably air bubbles in the transfer, where the actual protein didn't transfer. So this spot that's considered a negative in this protein band was really an air bubble that didn't allow the protein to transfer. So there's no data here. Okay? Now, is that fraud?
No.
It depends on if the student's doing it and the student's had very little experience with gels and doesn't know that much about air bubbles or whatever, it probably doesn't come to the level of fraud. It's just sloppy. On the other hand, they should have repeated this multiple times, and they should not have gotten that air bubble at the same place every time. But again, what they're showing is representative data, meaning the data that looks the best for them more often than not. So this is just an example to show you. And this is a case where the student prepared the figure using the data. He was responsible for it, and then only showed this to the PI. Now, as a PI, I think the PI should have looked at some of the raw data too, especially when it looks kind of weird like that. And is it fraud? No. Here's another example I've seen, and this is another one of these figures that got published. It's got bands. We tracked down in the notebook that one set of bands was here, another set was here. There's a couple things that are wrong here. One is, as much as possible, you want to minimize parallel experiments. Okay, and so when you do a northern blot, you could run all the same samples 4 times on gels, plot them, and hit each one with an antibody, or you could run them down one gel and sequentially put antibodies on one at a time, so it's the same membrane that's getting probed. And if you think about it, if there's a bubble in one of the gels that makes the sample run wrong, or if you load it poorly, or if you make a mistake and put the lane loaded in the wrong order, the more parallelizing you do it instead of sequential labeling, the more chance of an error. And in this case, what I really don't like is it looks like the gels were labeled or were put together in different order. I'm not even sure there is a lane here in this part that was shown. And if it is, that would be this lane, and then there's another lane beyond it that I don't see here. And so there's just a lot of stuff that made me nervous. This would be nervous enough that there may have been fraud on the student's part. Again, the PI sees this. It's not fraud, but he didn't take care looking at the student's data enough, okay, guiding the student. Any questions on that? Any thoughts? So part of what you do as an experimentalist is think, how can I make my experiments have the lowest variability, best control? Do I have any more? No. Okay. Last thing I'm going to cover. Am I out of time or anything?
Yeah.
4 minutes.
4 minutes. That should be just about right. Bayh-Dole is an act that allowed, was meant to encourage commercialization. It means that the NIH, even if they fund a grant, doesn't own your result. Okay? That's a cool thing. So now the university usually retains title to it. There are a few exceptions to that where with other funding agencies and so on. But basically, if I make a amazing discovery that gets commercialized, Tulane owns it. The nice thing is Tulane's very generous about sharing the revenue with me, but they are the ones who actually own it. There's a bunch of stuff on these websites. I think you guys all have copies of my slides where you can find out more about regulations and policies about any of this stuff. And then who owns the raw data and all the research? Tulane owns it. They own the raw data. They own your notebook. Whenever I've left a lab, my notebook stayed there. I was allowed to Xerox and make copies of my notebook so that I could look back and help write papers and all the rest of it. There's nothing wrong with copying data, but you can't mess with those raw notebooks. And I forget, I think it's 5 years minimum that you have to keep them, but basically I could show you my notebooks from certainly when I was a postdoc in Sanger's lab. I know I keep that one around. So ever since then, and notebooks from all my students, big, long shelf full of them. Now, it's my job as a PI to manage that data for Tulane. I got to keep those notebooks, okay? The only other thing I wanted to say, when I was thinking about this just before coming here, I was looking through my slides, and this is about data sharing. The other thing I just wanted to mention is there are other rules about sharing with your colleagues. For instance, when you publish something, you publish that you made a transgenic mouse that has some properties. Once you publish something, you're really pretty much obligated to share that. Okay? You're obligated to send somebody a little aliquot of your antibody. You're obligated to basically, to the extent you can. Now, sometimes this reagent is in really low short supply and you can't do much, but you're obligated to send your DNA clones and all the rest of that to people. Most of the journals absolutely require it. It's generally the accepted practice. There's databases out there for all your DNA data. The nice thing is once you put your DNA and RNA and so on data or any data into a public database, it's now accessible, you've shared it, and you don't actually have to back it up quite as much as you used to because now it's their job to back it up. So I'm always relieved when I've dumped my data into the DNA database because it was taking up terabytes of hard drive. And I've got all these hard drives around trying to figure out where the data is, digging back through them all because they're not always even mounted on the computer anymore. So just wanted to bring that up with sharing. Any questions? Anybody been unethical lately? Good.
You want names?
No, but Lisa does.